Hi, this is Regina Ragg from the University of South Carolina Center for Science Education. I wanted to compile a brief video of all the things that you could not see because you're not here in the lab with me. And they're all things that involve how you're going to get results from this unit. So, in order to get your DNA, I had to add what's called proteinase K to your saliva sample that you added DNAsol to. So, this has the proteinase K in it, and I let it incubate overnight here on my lab bench. Now I'm going to take 750 microliters of your sample, or 3/4 of a milliliter, using a pipette, and I'll add it to a microcentrifuge tube. Next, your samples are centrifuged at room temperature for 15 minutes at 13,000 RPM, or revolutions per minute. During centrifugation, the DNA is separated from cellular and other debris that is in your saliva. So if you look at the bottom of these tubes, you can see those little dots. That's actually everything from cell membranes and organelles to what you had for lunch. The next procedure step requires me to remove the supernatant with the suspended DNA from the old tube and transfer the supernatant to a new tube. Now I'm going very slowly and carefully because I don't want to disturb that pellet of junk at the bottom of the tube. That pellet contains all the material that we don't need in our sample. So I'll keep the new tube with the supernatant and discard the old tube that has that pellet of junk. DNA is a polar molecule because of its phosphate backbone. The supernatant right now is primarily water, and you know water is a polar molecule too. So based on the principles of like dissolves like, the DNA is dissolved in this solution. By adding 100% ethanol, which is much less polar than water, I'm able to precipitate out the DNA. After adding ethanol to the tubes, I allowed them to sit at room temperature for approximately 10 minutes. This is enough time for the DNA to fully precipitate out of solution. Next, I centrifuge the tubes again. This time, I'm going to discard the supernatant and keep the pellet that's lodged on the side walls. That pellet should be our isolated DNA. Now, I'm very carefully washing down the walls of the tube with 75% ethanol. Yet again, I'm going to discard the liquid and pipette away any excess, being very careful not to disturb that pellet. That pellet is your precious DNA. The very last step of the isolation is to add 150 microliters of DNA-free double distilled water back to the tube. After allowing this tube to sit overnight, the DNA will redissolve in the water, again based on the principles of like dissolves like. Tomorrow, I'll be able to actually conduct the next set of procedures in the PB92 taste receptor analysis with your isolated DNA.